All right. Hi everyone. I hope you are all doing okay and staying safe and staying healthy and keeping your heads above water. So we're gonna go ahead and start talking about the next steps for the class. Believe it or not, we are actually kind of starting to round the corner into the home stretch of everything we have to do. Today we're just going to do kind of a quick recap, focus on things that you should be working on this week, and then we're going to look ahead to the discussion section. So this is all posted on the Lab 10 overview, but just to kind of put it in another place for you, at this point, you should have done, or at least, attempted your analyses for all of your hypothesis testing and everything else. You should have done that all individually in SPSS and sent me that output already. That was due on Friday. And if I received that from you, I should have either given you the okay saying it's all good or asked for just, a couple more tests or some tweaks or something like that. Um, from what I've seen so far, between all of the group members, I think just about every group has everything they need. So I think even if, I requested more tests or edited tests from you specifically, someone in your group has what you need to work on the results section, which is the main thing to do this week. So the results section is due Friday by midnight. This is a group project. You need to have at least one figure as part of this, remember? So that would be like a graph showing your data. You can include a table or tables, plural, if it's going to be helpful for you, but that's optional at that point. But you do, just by the sort of the requirements of this course, have to have at least one graphical representation of your data. So the results section, the written results section, and the figure is due Friday by midnight. And again, that's a group project. Also, hopefully you saw my announcement, but just to remind you or to reiterate, this week I'm moving my office hours to Tuesday afternoon from 2 to 4. It's the same link that you have been using for the last couple weeks in that Google Calendar invite. And then next week I should be able to move it back to Wednesdays from 2 to 4. Of course, if these times don't work with you, let me know, right? If it turns out you have a class that you actually have to sit and listen to a live lecture for or something, let me know. We can find another time to meet. My time is pretty flexible these days. Okay, so let's go ahead and start talking about the discussion section. So this is the last section of the paper, believe it or not. So let's talk a little bit about what this entails and kind of some tips for getting it done. So the purpose of the discussion section is that it's kind of the denouement of your paper, right? You are interpreting your results that you just laid out for your reader. You're talking about what the implications of them are for the scientific community or for the broader, world, broader society. And then what can be some of the future steps that you can take from this data, as well as what are some of the limitations of your study that the reader should be aware of. So this section is essentially almost a mirror image, or it kind of goes in the opposite order of the introduction. So just like the introduction, I was telling you to think of it as an upside down triangle where it starts broad and gets narrow. Now for the discussion, it's going to be a right side up triangle, or just sort of a regular triangle, where your writing is going to start really narrow and then throughout it's going to get more and more broad to the end. So what does that mean sort of in practicality? Well, you're going to start again at the narrow point at the top of the triangle at the beginning of your discussion. You're going to be talking about and reminding your reader what your hypotheses were, right? It's been a minute since they've sort of read about your hypotheses. Just remind your reader, what were you expecting to find? And then talk about whether or not your hypotheses were supported. Remember, we're not talking about them being proven in any way, just whether they were supported or not. Then you might want to sort of situate your results within the literature, saying, how does this compare or contrast with findings that other researchers have found on similar topics? You're going to want to talk about what some of the limitations of your study were, what would be things that you might improve next time. And then finally, you're going to talk about sort of the broader implications of your work, right? How can this be applied to different contexts? How can this be applied to solve a cultural or a social dilemma, right? What can we sort of take away from your work? This is what we call the so what of your research, right? Like I read your whole paper, I see your findings, and I go, okay, so what? And this is where you tell me, well, it's important because blah blah blah. So this is the broader implications are the so what of your work. Okay, so a couple sort of tips for writing the discussion section. First of all, a lot of you did not find significant results. That's fine. And keep in mind that if you didn't find significant results, that doesn't necessarily mean that it was your fault or that you did something wrong, and you should still try to interpret the findings anyway. Sometimes null results are really interesting. You can also think about, were your results not significant because of something that has to do with your measurement? Like, did the construct or the scale that you were using, does it maybe work differently among, UConn college students, for example, or in this setting maybe? Is the situation that you set up, is it different enough from past studies that it might change the results? Maybe you were using a certain test and other people used different ones, or you were using certain stimuli and other people used videos instead of photos, whatever it might be. Kind of think about why you might have gotten these null results that conflict with what past research has found. And then you can also just think about, is it a true null, which just means, is it actually that there's just no effect between X and Y in the population? And if that's the case, well, then that can be really interesting too. So these are all things to be thinking about, and about how to discuss findings, even if you don't get a significant result. The important thing here, though, is to at first interpret your results before you talk about any kind of potential quote unquote problems in your study, right? So if you don't get significant results, this isn't the time to just sort of, talk about how bad it was or kind of like self-deprecate in that way. Still kind of professionally and scientifically interpret these results with a sense of objectivity, before you kind of go into what the limitations would be. You really want to make sure you're thinking critically about this topic. You're not just sort of restating what the results are, right? And by critical thought, I mean thinking about what some of the generalization of these findings could be. So, for example, how would this generalize, or what would be the external validity if we think about this spreading to other populations, maybe other settings, or, do your results or does your study kind of offer potentially a solution to a broader problem? Or could it help solve something, that's weighing down society? Or does it have implications for, a bigger social issue? These are the kinds of critical thought that I'm talking about here. So again, not just restating all of your results and, kind of relisting them, but talking about what they mean in a broader context. Now, when it comes time to talk about the limitations of your study, I want you to think really hard about them. Every study has limitations. Many people, including everyone in this class, but even most sort of professional scientists, are going to be limited by things like sample size, time, resources. That's sort of a given in a lot of scientific papers. And although we always do our best to either accommodate for those limitations or try to work around them, it's kind of always a problem in sort of the difference between kind of what we would ideally like to do in science and kind of the reality of running science. So when you're thinking about limitations for your study, try to think about things that are really more specific to your study specifically and wouldn't necessarily apply to just any scientific study. So for example, like, yes, you were limited by class restraints and the whole setup of this class, but again, the whole point of this paper is to practice writing a scientific paper. So, try to pretend this wasn't a class project for the purposes of your write-up, especially in this limitations setting or section. You do need to have at least 2 limitations listed in your paper. You can talk about a low sample size, but that's not going to count as a limitation. So 2 other limitations besides small sample size. That's just, you're going to have to get a little more creative. Okay. So the discussion draft is due on Monday. Sorry, that's not actually next Monday. That's actually 2 Mondays from now, April 20th by midnight. This is an individual assignment. I am once again offering a soft deadline for this. If you want to send me something to look at ahead of time, even if it's just an outline or half a draft or something, that's fine. Send that to me by this upcoming Monday, the 13th, and I'd be happy to look at it and give you an extra round of feedback kind of for free. There's instructions on Husky CT, there's the actual assignment, there's a rubric, there's tons and tons of resources on Husky CT, including example papers and tips on how to write discussion sections and all that. So really make sure you kind of troll through all of the writing resources on Husky CT. And this is your last paper draft. So after you get this done, you are done writing drafts of your paper, and then it's just the final paper at the end. So you really are getting close to the end here, which is pretty exciting. Okay, if you have any questions about the discussion draft, kind of what goes into it, how you should format it, etc., as always, you can email me. You can always post it to the FAQ thread on Husky CT so that everyone else can see your question and the answer as well. And then you can come to my virtual office hours. Again, this says Wednesday, but this week it's going to be Tuesday from 2 to 4 p.m., but normally it's Wednesday. So this week it's Tuesdays for virtual office hours. Okay, everyone. Stay well. Email me if you have questions. And I hope you are all keeping sane and doing okay. All right. Bye, everyone.